afternoon. I'm Linda Klavinskis from King's College London and a fellow and member of the ISV board. And it's my great pleasure to congratulate and welcome Professor Xavier Saelens from the University of Ghent in Belgium on becoming a fellow of the International Society for Vaccines. So Xavier, as a new fellow bestowed by this esteemed biomedical society, our membership and registrants at this annual Congress today will be interested to learn a little bit more about yourself and your scientific journey to this point. So let's start, Xavier. Can you introduce yourself to us?
Thank you, Linda. My name is Xavier Saelens and I work as a research professor at Ghent University and VIB in Ghent, Belgium. I obtained my PhD in biotechnology under the supervision of Walter Fiers, and my research in Ghent focuses on respiratory viruses, including influenza and human RSV.
So from working at the University of Ghent, how did you become associated? How did you get involved with the ISV then?
Yeah, well, since a considerable part of my research is on vaccine development, I got involved into the ISV activities early on, since the start of the society, more or less. And I attended several of the ISV conferences, and I'm very pleased and proud to have been invited speaker at the 2015 Congress in Seoul, in Korea, back then organized by ISV.
And so I know that you've been very modest here because your involvement has been considerably more than just attending some of the conferences. So I believe you're also a member of the board, is that correct? And can you tell us a little bit about your involvement there?
Yes, so indeed I am a member of the board since 2020. And I help to oversee the financials of the society and try to contribute to organizing events such as the COVID series, where I was a chair in one of the sessions. And I think my most visible contribution to the ISV has been when I co-organized with you, Linda, and Ted Ross, the meeting in the Society Congress in Ghent in 2019. Turned out to be the last live Congress of the ISV Society. And that was quite a bit of work and was, yeah, quite a journey as well. So I got involved in attracting funding, proposing speakers, selecting abstracts. I also remember very well that I, together with Sean Lu, was putting up the poster stands on a Saturday morning. So it's a very exciting time for me. I enjoyed that organization very much, and I think the meeting was quite successful as well.
Oh, yes, I do remember that very much, indeed, co-organising it with you. And I think it was actually one of the most successful ISV congresses we've had in the last 5 or 6 years. And that's thanks to all your huge efforts and your background team behind you in making it such a success. And we all enjoyed visiting Ghent. So let me ask you, becoming a fellow of the ISV is a huge honour. And in my case, when I became a fellow, I thought of it as a wonderful recognition of my contributions to vaccine sciences and to ISV and my standing as an immunologist in the field. And so, Xavier, what does becoming a fellow of the ISV mean to you personally?
Well, I feel very privileged and very honoured to be an ISV fellow. Because it indeed shows the appreciation of the society and the international peers for the work we've been doing in my lab on vaccine development and vaccine mechanism of action studies we do. So it's really a pleasure to be ISV fellow for me in the coming years now. And I hope I can, in that position, contribute to exposing, helping the ISV get a global view on its endeavors in the vaccine field, and help to use my network here in Europe to make contributions to the society.
I'm absolutely delighted that you've joined us and you're one of our fellows. So just moving on a little bit, it's always interesting to hear a little bit about the background story of distinguished scientists. And so first, can you tell us why you became a scientist in the first place? Because different people have got different reasons for becoming a scientist.
Yeah, ever since I was a kid, I was very much interested, and still am very much interested in nature, but at the same time also technology. And I was inspired by those two aspects of our world, let's say. And as a scientist, one has to be curious. So I wanted always to create tools to fix a thing, to help solve a problem. And when I was young, biotechnology became really a booming field, and biotechnology is so combining nature and technology in one discipline.
So specifically, what then drew you to the field of virology and vaccine sciences from all the different kind of areas of technology that, what you were interested in when you were, going through school? Was it really planned to become a virologist, or was it just a serendipity by accident?
Yeah, it was maybe to some extent planned with my master thesis work in the beginning when I was first in the lab here in the university. But virology and viruses have fascinated me a lot. I think virology is one of the most exciting fields in research, because those tiny life forms are so clever, evolved so tremendously evolved, so to the perfection almost, to make occasionally sometimes people very ill. So how they manage to do that is really intriguing. And then one next step is to try to do something about it, to learn from how a virus takes over an organism, elicits immune response, how we can learn from that to make a vaccine, understand how it works, and then again how to try to predict or solve how the virus may eventually escape again that vaccine. So that combination of these tiny creatures making us, making society, making a big impact on the societies. Think about SARS-CoV-2, for example, and how we can learn from that and put technology, biotechnology to our hand to fight those tiny organisms.
So when you first started, did you work on influenza virus? Because I know in recent, in the last year, like a lot of people, you've been working on also on SARS-CoV-2. But have you throughout your career always worked on influenza virus, or have you worked on many different viruses?
I have worked mainly on influenza viruses, although I must say I did my PhD on plant viruses, a very nice pair of viruses. There was a helper virus, tobacco necrosis virus, and it had a satellite. And that combination is well, one sees that more often in also in other virus virus host combinations, let's say. And the idea was there from the beginning to take the satellite virus as a vehicle to produce a heterologous protein, a cytokine, for example, using the helper virus to, of course, to make that cytokine then through the plant satellite virus. So influenza has been my focus for the most in terms of the virus we looked at, but also recently respiratory syncytial virus. We've been working on that virus now for more than a decade, and some other pneumoviruses are also under the radar now. So let's say I've touched upon a few viruses, but just a tiny scratch on the surface of the big world of virology.
So that leads me really nicely to my next question to really ask you is, if you could put a little bit more detail in how your career developed, because you've mentioned you've worked on a number of different viruses. So perhaps you can tell us a little bit how it developed and the different labs you've worked in and the choices you made along your research path.
So my academic career is rather atypical because I always stayed affiliated with Ghent University after my PhD, which is unusual for an academic scientist, certainly in modern times. After my PhD, though, I switched topics a few times. So I obtained my PhD with this plant virology work, and then I worked on influenza and mainly work trying to produce recombinant proteins, hemagglutinin, the neuraminidase in yeast, in insect cells, and try to make vaccines back in the 90s then. But then after a certain strategic decision in the laboratory here made that the vaccine development work in the institute was a bit on the down burner. So then I switched gears and I went to quite different field, namely the field of cell death research, which was an eye-opener for me because it was very fundamental. Signaling was involved, complex things. So and I tried to give it a touch of virology as well. But I learned a lot from that topic switch, so cell death research. And then eventually I got an opportunity to get promoted in the field of influenza vaccine development. And that's how I became an independent researcher with a focus on virology, with a focus on a number of respiratory viruses, with application-oriented research for the most.
So it sounds quite seamless, your journey through science and your career, but did you actually really encounter any particular hurdles in your career? I mean, most people have ups and downs, but maybe you didn't. And how did you handle that?
Yeah, so for sure there are in the research world, let's say, and in the lab and in our daily life. I think in general that is a human, typically human, let's say. But so the most joyful times are, of course, when you get a paper accepted, when you have a nice result on an experiment, when all the controls fit and you see this one band or this one population of cells popping out, or when a project gets granted. And then the downside is when this does not happen, when your experiments fail time and time again, when your project gets rejected from peers, which is always painful, of course. So this is something one has to try to live with. And in the end, I think the most practical way to handle it, to try to handle is to kick the door and move on.
So from what you said, that your career has been predominantly at the University of Ghent, and it's obviously been in the academics sector. Was there a specific reason why originally you chose academia over industry?
That's a very good point. So I have been hesitating a number of times to make a switch to industry, I must confess that. However, the academic environment where one has, at least to some extent, the choice or the freedom of choice to pick out the topic of one's interest was always appealing to me. And also the teaching duties and the teaching one's research to younger students is something I like a lot, and I would like to passage my let's say the findings in the lab, the way research works, over to the students, even if it's a general course in virology that I teach. So that attracted me a lot, teaching to younger students, and also the relative freedom of choice for one's research topic and priorities.
Do you find it now a bit of a tension to be able to manage both the teaching and, maintaining a very high-profile research career?
It is indeed challenging at times. So the academic year is about to start again here in the fall of this year here in Ghent, and one has to prepare for the classes, and this takes time, but then also it's rewarding, and it's you have to try to get the attention of the students all the time, and I think it works best if one can speak out of a passion, at least for certain aspects of the course where one is oneself involved. So at times I think the balance should be right. I'm more a research professor, but I do enjoy the tasks of teaching as well. So it's challenging, but I think I can manage it quite well for the time being.
So today, as opposed to maybe 20 years ago when we both started out our careers, research is now much more interdisciplinary, and it's much more focused on rapid translational benefit, for which partnerships and links with industry are enablers to clinical development. So can you tell us, in general terms, do you have involvement with industry, and how has that been beneficial to your research programs that you've been involved in?
So our research has always been on the applied side, so by looking at pathogens, of important human pathogens, and then trying to make vaccines or antibody-based therapies. One is quite close to an application oftentimes, and this occasionally would attract industry attention. And that is always, I've seen this always as an opportunity, because I like to keep an open mind there. The industry, of course, has its own interests to pick up a certain technology from a research lab, so that there's always there should always be a common interest, which I find is easily identifiable if you start to negotiate with an industry. I have been working with industry now for I think more than 2 decades, and again the field we work on is open to that, is amenable to that type of interactions. I think it's rewarding, and you start to appreciate what priorities can be for a project that you do with industry. It's different from an academic or a PhD project that one does completely in academic terms. But I also see it as an opportunity to train PhD students who have sometimes an interest to pursue their career in industry, then that they are already exposed directly in other ways to industry related projects in the group. So I like that opportunity when it's there, and when there's mutual benefit to collaborate with industry.
I think you're obviously in Ghent very fortunate to be in, particularly at the VIB, in that sort of environment that really encourages that interaction with industry. And am I correct in thinking that you actually have co-founded a small little biotech company?
Yes, that is correct. So I must indeed say and confirm that our institute, VIB and Ghent University, are quite supportive of industry-related research and industrial collaborations in general. But yes, right, with the COVID-19, we discovered an antibody, so an antibody-based therapy potentially for human. And that was a serendipitous finding that related to some intellectual property. Yeah, let's say we found a niche there. And that was, given the problem that COVID-19 is clearly to the society, was a strong enough file to start a company, which was supported by our university, by VIB, by private investors. So I'm a co-founder there of a company called Xavier Bio, and I try to help them in their startup, which is of course in biotech always a very yeah, it's difficult and exciting, challenging for startups in biotech to keep their financials right and their strategic plans up and running as they should be.
So I'm aware that you have a very large, successful laboratory, and that the antibody therapy that you've been discussing is just one aspect of the work. So can you tell us a little bit more about what the rest of the focus of your laboratory is?
Yeah, so we work on influenza, and that part of the work is mainly oriented at improving the influenza vaccines that are on the market now. So they can do better, and we try to contribute there by looking at one of the at the neuraminidase, essentially, so another antigen of the virus that is important for protection, we think. We also work on RSV, SARS-CoV-2 and influenza again, but then with the purpose to use nanobody-based technology. So nanobodies are type of antibodies that have been discovered here at our institute, to use these to try to prevent but also treat disease caused by influenza, by RSV, by SARS-CoV-2. So that has become a fairly large part of the work we do, and of course we want to then understand how these nanobodies work. Can we produce them? Can we make the best out of them? So formatting them up to the point that they are almost ready for clinical purposes. But apart from that applied research, I also have always tried to keep a fundamental research line underneath that, which is looking at, in our case, I have 3 PhD students who are working on the field of virus-host interactions, and that is protein-protein interactions, where we try to pick up new interactions, try to see how a virus fights a host response and vice versa. So the applied research is there with vaccine development, antibody-based therapies, and an important layer of fundamental research. So we try to keep the balance there.
So it seems as if it's all rather interconnected, and each all forward the other. Looking back, what do you think of the work you've been doing over the last 20 odd years? What do you think is the most surprising or unexpected thing that you discovered?
Yeah, there it's actually the recent work on SARS-CoV-2, which was really surprising. First of all, the event surprised the whole society in 2019, beginning of 2020. But also it was this nanobody I talked about, which was also one of the reasons for this startup company. We started to work on SARS and MERS in a side project together with Barney Graham and Jason McClellan in 2016. So it was really a side project, and then we stumbled upon this nanobody that happened to be cross-reactive, cross-neutralizing with SARS-CoV-2. So that was really something one couldn't plan in advance. Far from that, it was a complete surprise, a happy surprise, and hopefully a useful surprise for the COVID-19 patients.
So I know you're very modest, Xavier, and so this next question might be just a bit embarrassing, but can you tell us what you think is your most significant research accomplishment, what you've contributed to society?
Well, yeah, it's of course teamwork, but if I look back at the past career, I would say the contribution to the universal influenza vaccine based on M2E, I'm very proud of that, and especially that we discovered how this vaccine actually works in vivo, because that was for a long time unclear unclear how this vaccine could work, because in nature there's no immunity against this M2E protein. If you induce it, there is protection, and then we figured out that it's through an Fc-dependent mechanism, which was an eye-opener and a clear result that I'm proud of.
So, looking back at your career and reflecting on it, for some of our early career researchers who are just beginning their careers in science, can you tell us what lessons you've learned in developing your career and what tips you'd give them?
It can be hard in science, so don't forget that. If you want to get somewhere, you have to be able to be flexible and see opportunities. But for the most, I would say follow your heart. What is really your top interest in your career? So do you like the bench, prefer the bench? Then that is what you should try to make the best of. If you prefer writing or organizing experiments and supervising things, then you should probably follow that path. In an experiment, I think I have to repeat this all the time, and I emphasize it all the time in the lab here. Try to have your controls in place. The more, the better. It will always be the end question you get. What is the control? So think about a perfect experiment every time, every day of the week. And if you are in need for new knowledge in a particular field, try to knock on the door of the best people in that field, and there's a lot of chance that they will happily answer and help you with your question. That would be my advice to the young people. Follow your heart and do what you can do the best, and try to improve that even further.
That sounds like very sound advice. So lastly, Xavier, life, of course, is not all about work. And can you tell us what you like to do when you're not in the lab? So what are your outside interests? What do you do to relax?
Yeah, so there is not too much time outside the lab, except then for going home and take some rest. But I do like to read a book, and occasionally I watch Formula One racing on telly, that is. So that is something I like. Technology again, probably, and is there. And to relax, I take a walk and do some work in the garden together with my family, with my wife. So that's relaxing for me.
Very last question is, what is on your bucket list of interesting and fun things that you still plan to do outside work?
Yeah, probably a train journey through Europe, but without a face mask.
Yes, I can agree to that. So thank you, Xavier Saelens, for a very enjoyable Meet the ISV Fellows conversation. It's been an absolute pleasure for me to record this conversation, and I'm sure this will be have been equally enjoyable and informative to our audience. Thank you so much.